I come directly to the next speaker. It's Professor Kostas Kostarelos. He's currently the chair of the Nanomedicine lab at the Faculty of Biology, Medicine and Health in Manchester, and at the same time he is the chair of the Severo Ochoa Distinguished Professor at the Catalan Institute of Nanoscience and Nanotechnology in Barcelona. I'm very glad that you have agreed to come here now to see from the safety now to the application. Please, the floor is yours.
Peter, thank you very much. Beat, thank you for dedicating a session on graphene and 2D materials, even though, as we can see, it remains quite exotic for this audience. I just want to give you a bit of an update, really, on how we have managed in the last 10 years to translate clinically graphene. And even though you think, and maybe this audience thinks that the material is not really suitable for medical applications, I will demonstrate, I hope, that we have managed to push the envelope towards that direction. As we know in this space, in order to transform nanomaterials in next generation medical technologies, what we're aiming to do is essentially taking materials, advanced materials, into the clinic. So creating a clinical program around novel nanomaterials has been essential in the case of two-dimensional materials such as graphene, but also you see it in many other materials. I mean, think of quantum dots that got the Nobel Prize last week and still have not found really a clinical use yet. I will try to give you a snapshot of the clinical, of how we push the clinical translation effort with graphene materials. But before that, I just want to give you a little bit of a landscaping, very, very briefly as time allows. When we started this program back 10 years ago, as Peter mentioned, the carbon nanomedicine landscape was dominated or being occupied by discussions and applications of fullerenes, carbon nanotubes, and the graphene was only a kind of a newcomer in that space. And there were lots of different applications as potentialities. However, there was almost no in vivo study. I'm not talking about clinical, I'm talking about in vivo study done with the material back then. And the effort that we decided to undertake for many years was to have a bottom-up approach and try to design medical grade, what we call medical grade graphene oxide materials. These are highly water-soluble materials with extremely well-characterized surface and structural properties, oxidation degrees, and very careful attention to endotoxin impurities. And we also attempted to control with minimal batch-to-batch variation, parameters such as the one you see here, lateral dimension. At the same time, and through the funding of the Graphene Flagship Project, we attempted with multiple papers, as you can see here, to determine the preclinical safety profile and safety limitation profile of these materials by performing chronic longitudinal dose escalation exposure studies. And as you can see here from this transcriptomic result that I have only time to share with you today, we already can see that one of these materials, the very large graphene oxide sheets, these are micron large graphene oxide sheets, but extremely thin. We're talking about thicknesses of 1 to 2 nanometers. Were already giving us some kind of indicators that they are high-risk materials. And if you look at the histological indications of that or expression of that from our chronic studies in comparison to multi-walled carbon nanotubes, which was our positive control you see at the bottom here, you see that there are some remnants of the response we were getting with the high-risk carcinogenic multi-walled nanotube types from large graphene oxide materials. And through this work, we decided that if we're going to run a clinical program, we should exclude these large sheets from this. And we're trying now to integrate this knowledge into any advisory that will come out from regulators or the European Commission around the use of these materials. So I will focus and spend 5 minutes on each of the clinical trials we've run in the last few years, But before that, I want to emphasize and I want you to appreciate that back in 2015, the challenge for us was to envision and design a clinical translation program around graphene materials, but at the same time, which can be easily done, and at the same time, the funders, such as the European Commission or the UK Research Council that has been funding this type of work, were giving us 7 years to deliver. So I will take a breather here and emphasize 7 years to get an advanced material from the lab to the clinic. It's not an easy task. So what we've done is have 2 studies. We decided to go with 2 studies, which have now almost been completed, as far as we are concerned, have been kind of completed. I will explain to you why at the end of this presentation. The first I will share with you is a first-in-human graphene pulmonary exposure clinical investigation that we performed with the materials I showed you. And the aim of this clinical study was to basically de-risk the material as much as we can. So all of our endpoints have been acute responses to exposure to these materials. So let me take you through the study very briefly. This is a study that was performed. This is a proper clinical trial. As you can see, it's listed as a clinical trial in clinicaltrials.gov. And what we did is we used this chamber that was brought over to the hospital at the University of Edinburgh from the Dutch Public Health Institute, which basically consists of an aerosolized process where you put pollutants, possible pollutants, and these are inhaled by a face mask by healthy or patient volunteers as they are cycling. And that cycling element was intentionally designed in the study because you wanted to obtain 2 mimics, basically. One is conditions of exposure on the road as you cycle, and second, of course, to control the cardiopulmonary responses you're obtaining under relative stress, and for each individual, of course, that had to be modified and modulated. So I'm not going to have time to go through this design, clinical trial design. All I want you to take from this quite busy slide are the bullet points here. That was an investigator-led trial. We had a 2-hour inhalation exposure under cycling, and we were looking at acute cardiopulmonary endpoints. It's a single-site trial and a single-dose trial of 200 micrograms per cubic meter, which is equivalent to the very high-end dosing that you see from non-controlled studies, so occupational health studies, etc., that we were observing. And we recruited 14 healthy volunteers in this. These are the materials that we administered, that we asked the volunteers to inhale small graphene oxide and ultra-small graphene oxide. And the differences in their size are between a few hundred nanometers in the case of small and tens of nanometers in the case of ultra-small. Everything else was exactly the same, extremely thin materials, very well characterized. So what you see here, I'll go very quickly because I don't have time through the data, but I welcome you to go through the paper that will be published any week now, any day now in Nature Nanotechnology. You have acute cardiopulmonary function, and we looked at both the lung and the heart, that it's looking as if there is no reaction. And very interestingly, we looked at the systemic inflammatory markers, a bunch of markers using ELISAs, again, within the 0 to 6 hour time frame. So again, acute reactions. And you'll see, as you can see from the graphs here, we did not pick up any significant spikes in systemic inflammatory reactions. And very interestingly, we went and did very deep proteomic analysis of the plasma of those volunteers. And again, we see very, very few, less than 5 proteins of any type out of 1,000 or 2,000 proteins that we normally see when we have patient diseased versus healthy volunteers. In the proteome of these volunteers post-exposure, and what you can see here in these diagrams, and you can tell from the dot plots on the left, you'll see here that we have almost identical blood profile between the two materials, but also at the 0 and 6 hour time point post-exposure. So all looking good, a kind of a check clinical trial here. There is lots more data, of course, that we've done that you are welcome to read in the paper. Overall, we have not seen any acute reaction from the inhalation of the material from these volunteers. Of course, the study comes with a lot of limitations. One is a single dose. Two, we're not looking at chronic exposures. And three, we're only talking about one particular type of material that's very well characterized, very clear, not really reflective of the commercially available graphene oxide materials, I have to say. The second clinical trial is of a completely different nature, and this is, again, a first-in-human with a graphene-based medical device in this case. And in the context of this meeting, I have to say that I've been discussing with Beat and Patrick for many years that nanomedicine is a little bit bigger than pharmacology and drug delivery, and sometimes we forget about that. So I will try to bring a little bit of medical device engineering and clinical translation into the meeting in the next 5 minutes. The aim of this clinical trial was to demonstrate advanced material, in this case graphene oxide, proof of concept function and safety using a neural interface device. Neural interfaces have captured a lot of attention in the last few years for the wrong reasons, namely Elon Musk, if you follow the news. But in the context of the Graphene flagship project, which was funding this work, we decided to engineer and fabricate these types of devices that you see here in the schematic, and in the subsequent 2-3 slides you will see how the real devices look like. The whole concept was to use a combination of unique properties that graphene is offering: transparency, electrical conductivity, and stability of the material, chemical and structural, as current is passed through the device into the tissue, and as the device is able to record neuronal tissue. So we wanted to achieve multifunctional devices that were able to record neural activity on implantation in the brain or the peripheral nervous system, and of course stimulate the brain or peripheral nervous system for therapeutic purposes. We also attempted to build a therapeutic drug delivery element that you can see at the top of the slide. And of course, all of this under the umbrella of pure biocompatibility. I am putting this slide here just for you to get a feel of the type of multi-component engineering that took place into this effort. So basically, we're talking about very flexible neural interface devices that consist of only the contacts made of graphene. All the substrate is made of a polyimide or other polymer that has an established biocompatibility profile, because other devices are made of these. And what is very interesting, and I want to emphasize, is that our starting material of our contacts, of our graphene contacts, was again graphene oxide. So the same material that I showed you in the first clinical trial, which was a safety trial, pulmonary safety trial, here we decided to take on board, and through this kind of engineering and membrane formation, trying to create an electrode, a neural electrode contact that will be in direct interaction with the brain tissue or the peripheral tissue. Here are some of the designs. At the top, the cortical surface probes, as you can see, the ones that are placed on top of the brain. And these ones are some of our designs on deep-seated probes. These are the ones that we are implanting deeply into the brain and the peripheral nervous system. And what you see in black dots at the end of these contacts is exactly where the graphene is. And of course, I want you to appreciate that, A, we can fabricate many of those in one go using those masks, using 4-inch wafer technology that the transistor industry has optimized. And the second is that you have a great flexibility of those devices. I only have time to focus and show you how we push that into the clinic. And we really, before we did that, we ran a series of preclinical studies. One of those you see here, where we could identify that both in terms of impedance and signal-to-noise ratio, in benchmarking exercises, we could be at least equivalent or better than the state of the art. In this particular case, we used the neural nexus device for those of you familiar with this work. And that was, again, on-table electrostimulation and recording experiments of the brain tissue. And the overall message I want to communicate is that from a series of papers that you can find here, we have basically opened up the frequency spectrum using graphene in terms of what can be read by the brain tissue. We know now that we have the capability to record at frequencies that are way above the current state of the art and way below the current state of the art. And we're exploring these capabilities in terms of recording for detection of signals, for example, pre-epileptic seizure signals or post-stroke signals, in order to be able to allow the surgeons and the clinicians of those patients monitor the condition of their patients much more carefully. But how could we translate? We couldn't translate as academic institutions, and that's one of my take-home lessons for you this afternoon. We had to set up a company, or we took a deep breath and set up a company and prayed that everything's going to work out. So we put together this startup called In-Brain Neuroelectronics. It's based in Barcelona, but actually has engineers and scientists from across Europe. We were very lucky to receive, the company was very lucky to receive a very positive response from investors. It has raised over 25 million euros at the moment. And we focused at this upscale engineering exercise where we had to go about 2,000 times higher. All of our devices I showed you until now were designed for this brain, and we had to go here, which is about 2,500 times larger in scale. So these are some photographs of the devices we had for mice and rats, for our rodents, and this is where we had to upscale to, and that was done during COVID. So you can imagine the effort we had. So what is it we've done, and where are we right now? And this is where I'm going to close with. This is the clinical trial that's undertaken at the moment as we speak at Royal Salford Hospital. The device is the one you see here. It is going to be used as an on-table surgical tool. The surgeons, the neurosurgeons are using this during a glioblastoma resection surgery, and the whole purpose of this device is to be able to give them higher fidelity using the graphene contacts, both recording and stimulation, by which they will be able to have a much more careful and precise resection procedure. The whole trend in neurosurgery in this particular case is to push and do supratotal, what they call resection procedures. So they need to make sure that they are not resecting essential structures of the healthy part of the brain, but they're removing the tumor tissue as much as possible in order to leave minimal residual tissue, tumor tissue behind. This is a prospective interventional single-site trial. It's taking place in Salford Royal. It only focuses on adult glioblastoma patients that are undertaking resection surgery, and we have 8 to 10 patients, and the follow-up will be 6 weeks. We're hoping to complete the study in 2024, and this will be the first ever, we think, graphene medical device trial of this type, but any other type, even biomarker studies that are in contact with the human tissue. I'm glad to say that we got FDA approval about a month ago as a breakthrough device for the designation for this technology, which also helps a lot and accelerates the regulatory framework and approval of this type of technologies. And again, we're hoping that the company will be successful in their subsequent series of funding. I have a slide of lessons, but I will just go very quickly. I would say maybe that's a good way to open the discussion as well. I think in order to translate technologies with advanced materials first, I would say you need to have an optimal model of translation. There's no golden rule here. You need to play with the specific type and specific technology you're developing and determine what's the best translation, what's the best clinical trial you need to design. The second thing that our lesson, that our experience has taught us is that there's no optimum time over first in human. You're under pressure from investors, from colleagues, from funders to go very quickly into clinical trials. Sometimes it's not optimum for the exact reason I mentioned here, that if an early failure could be catastrophic for the technology. So you need to make sure you go into the trial when the technology is reasonably mature. And 2 more points. Industry, large industry particularly, does not wish to deal with academic institutions and research centers. That's for sure. That I have established throughout 30 years of doing this business, in this business. So I would certainly recommend starting up companies and letting those deal with the larger and bigger players in this space. And of course, a more philosophical point, definitely for our discussion, that I want to leave you with is everybody, when you have an advanced material, a new material, keeps asking about de-risking. But then the question is, how do you de-risk the unknown? Because there are not known risks for an advanced material. First, you need to identify the risk and then de-risk. So how do you de-risk the unknown? Our teams in our lab that have contributed to this work are the ones that should be thanked for, particularly Cyril Boussy, who's running the nanoinflammation lab in Manchester and has also participated in all of the preclinical and led all the preclinical work I showed you with the graphene oxide materials from the pulmonary safety point of view. And of course, the Neus Lozano, who has designed all of these materials that even Bengt, our previous speaker, showed, and is running the team in Barcelona at the Institute of Nanoscience and Nanotechnology. I want to thank Mark Miller and Fleming Cassie, who are the 2 key guys that allowed for the execution of the first clinical trial with the pulmonary inhalation studies, and Jose Garrido and team in Barcelona, who are the chief engineers. They've designed all of these flexible graphene-based devices I showed you, neural interface device I showed you at the preclinical level and a little bit at the, then the company took over. And of course, our neurosurgeon, who is really leading, and he's the chief clinical investigator of the glioblastoma resection trial that is running right now. Thank you.
Thank you very much, Kostas, for this tremendous journey through lab to bench.